I'm excited to share with you the results of a recent publication that we had accepted into Life Science Alliance. In this video, I'll give you a brief overview of the key methods we used and go over some of the main results from this study. As autism is neurodevelopmental, we were interested in studying the impacts of variants associated with autism in both fetal and adult cortical tissue. First, we used gene expression and chromatin interaction data to create fetal and adult cortical tissue gene regulatory networks. We looked at what SNPs, or genetic variants, spatially interacted with what genes, and whether these interactions resulted in a change in gene expression for that gene. This gave us a list of SNP gene regulatory pairs. We then used GWAS data to subset those gene regulatory networks to only include the SNP gene interactions for the SNPs associated with autism. Next, we extended this out using protein interaction data. We identified the proteins interacting with the proteins encoded for by the genes regulated by autism-associated SNPs. Eventually, what we ended up with was a network of protein interactions, the genes that encode those proteins, the SNPs that regulate those genes, and finally, we looked at the GWAS traits associated with these SNPs to identify the genetic overlap between autism and co-occurring traits in the fetal and adult cortical tissue. What we found was an association with a large number of brain and mood-related traits. We also found some non-neurological traits, such as lung traits and BMI. However, we found a particularly large overlap with schizophrenia. Overall, there was a range of neurological traits and conditions identified, such as depression, mood-related traits, and Parkinson's disease in both the fetal and adult cortical tissue. In just the fetal analysis, so those traits that didn't appear in our adult cortical tissue analysis, we identified traits related to brain development. Looking more closely at the fetal analysis, we found distinct clusters of gene trait pairings. So what this figure is showing is that there are SNPs regulating this gene, for example, associated with autism, and SNPs regulating the gene associated with the other traits on the side here. This large cluster here shows that these genes here are all associated with the intersection between autism and many brain traits, such as white matter microstructure, Parkinson's disease, and mood traits. What's interesting is that all of these genes are at the same loci, chromosome 17q21.31. We also found another cluster associated with cognition and memory and worry, and this time, all the genes at this cluster are at chromosome 3p21.1. Finally, we found many genes across the genome associated with the overlap between autism and schizophrenia. Next, we did a two-sample Mendelian randomisation study to identify genes putatively causal for autism. Through this, we identified 4 genes putatively causal for autism in the fetal cortical tissue, all of which are at the chromosome 17q1.31 loci that I mentioned on the previous slide. In summary, we've found developmental windows such as fetal development, where genetic loci, such as chromosome 17q21 and 3p21, have the potential to impact on trait combinations. Within the chromosome 17 loci, we found genes putatively causal for autism. Thank you for listening. We hope you enjoy.